Hello friends, today we will study about making a gene map. In question this data is given. We have to find gene order, map distance, coefficient of coincidence and interference from this data. Then first we will see the first step of finding gene order is to see which is the highest 321 and 308 because linked loci tend to stay together. That's why the non-crossover or parental phenotypes are most frequent. Which one is most frequent? 308 and 321. The genotype is A plus, B plus, C plus and A, B, C. This is 321 and 308. The next step is we have to find which one is least frequent because simultaneous crossovers between outside and middle loci are unlikely, so double crossover genotype should be least frequent. The least frequent phenotype is this one and this one. A plus B C 18 and A B plus C plus. These are double crossovers and these are parentals or non-crossover. Then in third step, to determine gene order, we have to compare parental and double crossover phenotypes. The parental phenotype A plus B plus C plus and double crossover we have to compare this. The marker that appears to switch places is in middle. We already know that in double crossover the middle places is switched. If it is parental and it is double crossover, then if we assume this is gene, then middle should be, if double crossover will take place, then here C plus B plus A and B A plus C. So we find it, the gene order will be B, then A, then C, because the middle place is switched. This is middle which is switched here from A plus, this is A, so it should be in middle place. So the gene order is B, A and C. Then we have to find map distance. First we have to find single crossovers. Between B and A. Single crossover between B and A means here it is B, A, then single crossover between B and A is B, A plus C plus and B plus A and C. From the equation we can find this data: B A plus this is 102, this is 112. So to find map distance between B and A, the formula is map distance is equal to percentage recombinant number of progeny in single crossover plus number of progeny in double crossover, sorry, not plus, multiply into 100 upon 1000. So number of progeny in single crossover is 112 plus 102. Sorry, it is in case of coefficient of coincidence, but in case of this it should be plus number of progeny in single crossover plus number of progeny in double crossover. 112 plus 102 plus 18 plus 15, these are in double crossover, upon total progeny, then it is 24.7% of recombinant progeny, and this will 24.7 mu apart from each other in B and A, 24.7. Next we will find map distance between A and C. Then crossover, single crossover between A and C. The gene order is B, A and C. B plus A plus C plus and B, A, C. Crossover between A and C, the genotype is B plus A plus C, B, A, C plus. From the question, B plus A plus C, B, this one, 66 and this is 59. So we find map distance between A and C is equal to number of progeny in single crossover between A and C plus number of progeny in double crossover into 100 upon total progeny. Here 66 plus 59 plus 18 plus 15 15.8% or 15.8 mu. The map distance between B and C is 24.7 plus this one 15.8 is equal to 14.5. Then we will make gene map. This is B, A and C. This is 24.7. This is 15.8 and total is 40.5. Then we will find we are already find gene order B, then A, then C, and map distance also 24.7 between B and A and 15.8 between A and C, and we have to find this one coefficient of coincidence. The formula of coefficient of coincidence is the observed double crossover upon expected double crossover. Then observed double crossover, we can find it from the data, which is 18 plus 15. And expected double crossover, we have to find expected double crossover from gene map. We know that this is 24.7 and this is 15.8. Then expected double crossover is 24.7 percent and 15.8% of total, that is 1000. So it can be calculated as 33 .247.158. Then by solving this, we can find coefficient of coincidence. The formula is observed double crossover upon expected double crossover. Observed double crossover value we can find from the data, which is 18 plus 15, and expected double crossover we can find from this gene map and map distance we have calculated. This 24.17 plus 15.8 and directly from this, and this is the answer which we calculate from this is the coefficient of coincidence, and the interference is equal to 1 minus CC. Okay, thank you.